So we finished up chapter 9 with the bones and our joints and metabolic, not imbalance, homeostatic imbalances. And now we're moving on to muscle tissue. So we're going to talk about skeletal muscle mainly. But the end of the chapter, we will get into cardiac and smooth. And you'll see where some of the things correlate, meaning all three types of muscle have some similarities to it. And sometimes skeletal muscle is very specific, and sometimes same thing for smooth and cardiac. But there are things that are across the board the same for all of them, which is nice. And starting with skeletal muscle tissue, we have functions of the skeletal muscle. So some of the obvious ones would be body movement, right? We wouldn't locomote or get to move anywhere if we didn't have these muscles. We have some skeletal muscles that also help maintain our posture. And it's also for protection and support. And then also heat production. So heat production, 75% of our body heat comes from our skeletal muscle. So you kind of wonder why men tend to be warmer and you have the females that are like cold all the time. Men have more skeletal muscle mass, thus more heat production. For the protection and support part, that would be our muscles do help protect and support our joints, blood vessels, and nerves. So that's just some general functions of our skeletal muscle. As far as characteristics of the skeletal muscle, this also is not just for skeletal, but this includes smooth and cardiac. All three muscles, all three types, need to have excitability, which is that the cells respond to a stimulus, and that could be a chemical stimulus, it could be a stretch or mechanical type stimulus, okay? All three types of muscles have conductivity, which is electrical charge travels along the plasma membrane. And then contractility, which is muscle cells can shorten. And then elasticity, we've talked about before, and this is the ability to return to normal length. So we're talking about stretch and recoil, right? So this would be after shortening or lengthening. Oh, let's try to write. How about that? After shortening would be a contraction, right? Shortening or lengthening. And so we're talking about stretch and recoil with elasticity. As far as extensibility, that's lengthening of the muscle cell. And so you're thinking, well, that sounds kind of weird. But for example, if you wanted to contract your quadriceps muscle in your thigh, right? So if you wanted to contract it, the hamstring muscle has to relax and elongate a little bit in order for you to contract this one. Or if you want to talk about your bicep muscle, in order to contract it, your tricep muscle has to relax and allow you. If both are contracted at the same time, do you move? No, you're stuck. So one has to lengthen while the other one, the opposite one, is able to shorten. So that's the idea of extensibility. So that's with all three of them. That's skeletal, smooth, and cardiac with characteristics. For anatomy of skeletal muscle, we're really talking about the skeletal muscle organ, which means the whole length of the muscle itself, okay? So if this is the humerus, for example, in this picture, what muscle do you think we are looking at based on some defining features on that humerus bone? You have a tendon that's actually going through something that it's named for. Remember bicipital groove? That's the biceps muscle. Just saying. All right, so do you guys see your bicipital groove right there where the tendon of the bicep muscle is going through? Okay, so maybe help you guys remember some of your bone anatomy there. All right, so that's the bicep tendon going through the intertubercular sulcus or bicipital groove. The anatomical neck of your humerus is going through that area right there. This would be the surgical neck area there. There's a tendon at each end of your muscle, correct? And it is made up of mesiums. It looks like mysiums, M-Y-S-I-U-M-S. So we have mesiums. And there's a couple of words. So when we start talking about muscle, it's like learning a whole new language. So now you're going to have some terms that whenever you hear that term, you know specifically it's talking about a muscle. And some of them correlate to terms you've already heard before, but now you need to change your language and realize what part of the muscle that you're talking about. So mesozymes are like a connective tissue wrapper. You can almost think of it like cellophane wrapping around something, right? So mesozymes, you have an endomesozyme, you have a perimesozyme, and you have an epimesozyme. So these will end up coming together and end up making the tendons at the two ends of the muscle. Okay, so it's these mesozymes that end up being your tendon tissue, and that's where it will then attach to the periosteum of the bone. And the periosteum of the bone, what kind of CT are we talking about there? This goes back to your question from before, from the other day. About the knee, when you were asking, yeah. So dense regular CT. So why is it regular for a tendon? One direction. Okay, muscles going in one direction. So irregular allows for multi-direction pull. Okay, so when we're talking about like the fibrous capsule, okay, that CT is irregular because it may get stretched in different directions from a joint, but it's not, when you're talking about a ligament that's only in one direction, or you're talking about a tendon in one direction, you don't want it to be stretched in multi-directions because its job is, so let's say for the quadriceps tendon, this is all it's allowed to do, bend and flex the knee. So we don't want it to be able to stretch in other directions. It only needs to be dense regular because the fibers only have to run in that one directional plane, okay? So dense regular at each end. But where the wrappers surround the muscle belly, it actually becomes dense irregular CT. Am I trying to confuse you guys? I know, right? You're just going to have to memorize this stuff. It sucks. I know. I'm sorry. So the mesozymes around the muscle belly are dense irregular, but by the time it becomes the tendon, it becomes dense regular. Okay? So let's see. And part of it is, what do you guys think when you are contracting your muscle? Okay, I don't have big muscles, but pretend I have like a huge bicep muscle, right? So if my bicep muscle, when I contract it and I get that little bump up, what is my muscle doing when I'm contracting it? It's shortening, but it's also like pushing out in a certain area. So does it make sense that the mesozymes wrapped around the muscle belly needs to be able to allow for movement in multi-direction? Thus the irregular, dense irregular part. So multi-direction, dense irregular. But by the time it gets to the tendon, the attachment points for it, that's where it's only in one direction moving. It doesn't have to expand in the tendon. That's why it's dense regular when you get there, okay? And let's see. So things change depending on the location and the stress on the tissue or the function of it. And let's see. Oh, if you were to, I know this might gross some of you guys out, but those of you that are meat eaters, and it's a lot easier to notice it, say like on a piece of steak or beef versus like chicken. But if you were to cut your steak in half and go against the grain, right? It's supposed to be more tender when you cut against the grain, right? I'm not telling you to eat somebody. You can eat a cow. I like cow. So when you cut it, what you guys are seeing when you see those grains, you're seeing the mesozyme. You're seeing that perimesozyme because it's sectioning off multiple muscle cells in that wrapper, and that's how you're able to see it. So some of this you could see with your naked eye, right? You're not seeing the individual muscle cells within it, but you can see the bunch of muscle cells that are put together. That's what you're seeing. So let's see. So one fascicle. So the perimesozyme is surrounding a bunch of these. Then if we have the little sections within, that is, if we have one grouping, which is like this guy right here, or this guy right here, that is a fascicle, okay? One fascicle, which is a grouping of the muscle cells, okay? And then if we pull one muscle cell out of that, okay, then let me start over. One fascicle is a bundle of muscle cells. One muscle cell runs through the entire length of the muscle organ. So from tendon to tendon is one muscle cell. That's the whole length of that. So your biceps tendon, one muscle cell of the biceps muscle, sorry, not tendon, runs that whole length from tendon to tendon. Does that make sense to you guys? Okay. So then it's a long skinny cell. one fascicle is a bundle of many muscle fibers. And then the perimysium is surrounding many fascicles. Does that make sense? So the one muscle cell is surrounded by endomysium. Then you have many of those muscle cells grouped together, which is surrounded by perimysium. Peri means around. Endo is within, peri is around. And then you have many fascicles grouped together, which gives you and are surrounded by the epimysium. Epi means upon. So that's the most outer layer of CT wrapper, if you will. All right, so one muscle fiber is the same thing as saying one muscle cell. You guys are going to hear a bunch of AKAs, and you just have to memorize all of them. I'm sorry. So a muscle fiber or a muscle cell, and one muscle cell has a bunch of rod-like structures inside of them called myofibrils, which we'll learn about in a minute, and those are made up of myofilaments. So myo, M-Y-O, is another thing that tells you, if you hear the word myo something, you're always talking about muscle, okay? So M-Y-S for mesium means muscle, M-Y-O means muscle, and in a second you're also going to learn the term sarco will also mean muscle. So you have 3 AKAs for muscle. Okay, any of those terms, you know you're talking about muscle of some sort. So if we look at connective tissue sheaths, okay, so we had endomysium surrounds each muscle cell or muscle fiber, because we said muscle cell equals muscle fiber, same term, okay. And then perimysium we said surrounds the fascicle, so a fascicle is a bunch of muscle cells grouped together. And then an epimysium is surrounding or envelops the entire muscle. Epi upon, that's the most outer part. So, and at each end, the muscle tendon is where all 3 of those mesiums, epi, endo, and endo, peri, epi, come together to make your tendons, right? Okay, awesome. So if we look at blood vessels and nerves, so you guys know that muscle tissue is well vascularized, right? If you cut it into a piece of juicy steak, what do you see? I'm just kidding. I'll get off the food. I didn't eat enough lunch today, apparently. So blood vessels and nerves, our muscle tissue is well vascularized. Okay. And in general, one muscle organ, meaning like your bicep muscle or your tricep muscle. Okay, so I do little m for muscle for me. One muscle organ usually has one major artery and one or more veins, because you need to drain the deoxygenated blood back up to the heart, correct? So it can get reoxygenized. And each muscle cell has its own nerve cell that controls it. So that means it's voluntary control. Okay, so if you want to move your muscle, you're thinking about it and you say, I want to walk over there. I want to pick this up. You are deciding to do that versus our smooth muscle is involuntary. Cardiac muscle, involuntary. Because you don't want to have to think about your heart pumping. Don't forget to beat. Hurry up and beat. I'm going to go to sleep, but don't forget to beat. No, it has to keep going or you don't keep going, right? So skeletal muscle is voluntary control. All right, and if we talk about microscopic anatomy of skeletal muscle, now we're seeing the term sarco here. So sarcolemma, sarco means muscle, so this is the outer membrane. So, your, what's the other word I was trying to say? It's basically, this is just your special name for the muscle cell plasma membrane. So sarcolemma is just the plasma membrane of a muscle cell. So if you hear the term sarcolemma, you're just thinking muscle cell plasma membrane. That's all. Sarcoplasm, what do you think that is? Is the special name that we give for the cytoplasm within a muscle cell. It's just cytoplasm, but because it's in a muscle cell, now we're going to call it sarcoplasm. That's all. It also contains a lot of glycogen. Do you guys remember what glycogen was when we were talking about our various molecules at the beginning? This is the animal storage form of glucose. So we link a whole bunch of glucose, glucose together, and we store it as glycogen in our muscle cells. Okay. If I was a plant and I was storing my glucose together, I'd be storing it as carbohydrate. Okay. So when you're eating your big fat steak, you're eating a bunch of glycogen with it. Yum. Okay. I need to eat. Anyway, multinucleate. So embryonically, the stem cells fuse together and contribute their nuclei. So basically, in order to create this long cell that we have that runs the whole muscle organ, they used to be smaller cells that then fused together. Each cell had its own nucleus, but when it fused together, it kept the nuclei, but now it doesn't have the individual membranes in between them. So you end up with one long cell that runs the whole length of the muscle organ with many nuclei in it. And that's unique for skeletal muscle, which is different when we end up seeing smooth or cardiac. Okay. So let's see. That was awesome. Now, myoglobin. What does that sound like to you guys? Hemoglobin. What does hemoglobin do for us? It helps transport oxygen, right? So myoglobin is specifically in your muscle. So it's a specialized or special protein that stores oxygen in muscle cells. So it's just like hemoglobin. It works very similar to that. So let's write special protein that stores oxygen, or O2, in muscle cells. And let's see, myofiber. So remember we said before, one muscle cell is also a myofiber. So you might hear me just say fiber sometimes. And it's all saying the same thing. Myo is definitely telling you it's muscle. When we use the term fiber later on and we're in like nerve, then I'll talk about it that way. But for now, whenever we're in muscle section and I say fiber, it really means one muscle cell or one myofiber. So if we look at our picture here, so how long is one fiber? Length of the whole muscle, depending on what muscle you're talking about, right? I mean, the length of the muscle, depending on the length of that actual muscle. Okay. So the picture here is showing you that this is one myofiber or one muscle cell, okay? And then it's surrounded by what surrounded one muscle cell? No? Not the muscle organ, but one muscle cell. Endomysium, right? So we'll have a grouping of muscle cells that'll make up the fascicle, which will be perimysium, and then we'll have a bunch of fascicles together that'll then be covered by epimysium. So this is an excellent time when you guys, like off to the side, you could write in order how you would find it. Top to bottom, bottom to top, inner to outer, however you want to write that out. That's what I would do if I were studying for this part. I would chunk this up into sections, and I would go through and say, okay, this is covered by this, and I go from smallest to largest. Another way you could remember it, if you want, is you could say myofiber, then myofibrils, and myofilaments. Okay, so myofiber is one muscle cell. Myofibrils make up within the muscle cell. Okay, these are going to be proteins that we're going to talk about. And then myofilaments are the proteins inside of it. So these are a longer word, but it's the smallest unit of what makes up inside of the muscle cell. This is the actual muscle cell. So whatever helps you guys remember it. Smallest to largest, longest word to smallest word. Remember, it's inversely proportional, short to long, long, short, whatever works for you guys. But you guys have to do these kinds of things to help you remember when you get on the exam. Because all these myo words, they get confusing, right? I can tell you guys are looking at me like, Liz, really? Seriously? All right, so myofiber is one muscle cell. It ends up being covered by this here, which is the, what wraps around the muscle cell? What's its membrane called? Sarcoplasmic sarcolemma. Okay, so this is a myofiber. This is a sarcolemma, because remember our cells all have to have a plasma membrane around it. Okay, so they peeled back the plasma membrane so you could see the sides of the cell. And then we have one myofibril, okay, and that myofibril will have myofilaments within it, which make up the protein within there. Myofilaments are the protein strands that you're seeing. Do you guys remember the terms actin and myosin? I think we said actin before, maybe myosin, I can't remember at the beginning of the semester. So myofibrils, which is number 6, are long cylindrical structures filling the muscle cell. So myofibrils also run the length of the muscle cell. And they make up about 80% of the cell volume. Thus, all that idea of where you're eating meat, protein, lots of protein in it, 80% protein, not so much cytoplasm or sarcoplasm and things like that, right? And so the myofibrils display what are called sarcomeres that we'll talk about in a second. So myofibrils display, here's that term sarco, right? Which is basically it shows the banding pattern. Because what do we say that when you guys are looking at histology? What do skeletal muscle have when you look on the histology slide? Striations. This is that banding pattern. So we're going to go into a lot of detail of what makes up that banding pattern and how it changes. And you guys really have to get this whole myophysiology stuff down, because you will see it again throughout this semester, and we refer to it again in 228. And when you learn like nervous physiology at the end of this semester, it builds on what you already have learned for this. So after today's lecture and the next lecture, make sure that you guys go home, talk this out like a story. I'm going to mention that a couple times, so that way you get comfortable with not just the terminology, but that you can relate the story and know the order that things happen, and you don't get lost, because all these words start to sound the same, right? Myofibril, myofilament, myo something, sarco, right? It gets confusing. So let's see. You could think of the sarcomeres. So this is one sarcomere. The sarcomeres are set up, you could kind of think of them like train cars. So you have this long train. Each train section is a sarcomere that makes up the whole length of the myofibril, or one muscle cell would be the whole length of the full train. But within the train, it has all these little sarcomeres. And within the sarcomeres, what make up the banding pattern are what we're going to find out about on the myofilaments. Okay? And look, there's myofilaments, protein strands that make up myofibrils. So when you guys review this stuff and you're trying to understand the terminology, repeat every single thing over and over again when you say it. Don't just assume you remember it, but the more you say these words again and again, the more you'll get comfortable with it, and the more it'll make sense. At the beginning, you'll be like, I don't know the difference between this versus that versus that. But if you write it in order of like smallest to largest and what each thing is, and you repeat it like a story, and you're going to tell somebody about this, it will help you guys a lot. So myofilaments are protein strands that make up the myofibrils. We end up having a thick and thin filament, okay, or there's many filaments in there. The thick filaments are made from myosin and myosin ATPase. And if something has ACE attached to the word, it tells you it's an enzyme. And for most of our purposes, enzymes are breaking things down. So it sounds like we're going to break down ATP in order to be, you'll see in the physiology part in a little bit. But so the thick filament will have myosin and myosin ATPase. The thin filament is going to have actin, tropomyosin, and troponin. Typically, when we're just saying thick and thin filament, we usually are just mentioning myosin and actin. But I do want you guys to realize that the thin filament with actin is actin with another little molecule on top called troponin, and then he has like this little string that runs the length of his protein that is the tropomyosin. Yes, okay. So actin is two beaded strands that are twisted or intertwined together and it has a binding site for the myosin head. And I'll show you pictures of these guys in a second. So has binding site for myosin head. Troponin, which we said is part of our thin filament, is a string-like regulatory protein. So I told you it kind of looks like a string. String-like regulatory protein that covers the binding site when the muscle cell is relaxed. So if a muscle cell is relaxed, the binding site is covered up, meaning when you guys learn about the physiology, the myosin can't attach, okay? So when we actually contract or shorten a muscle, the myosin head will be able to attach. Tropomyosin will be out of the way so that myosin can attach. When we're relaxed, tropomyosin is covering that site so things can't attach to it and tug on it, if you want to think of it that way. Troponin is a globular, I kind of think he looks like a snowman in the picture in a second. Troponin is a globular regulatory protein attached to tropomyosin. It binds calcium, and my shorthand for calcium ions is Ca with 2 pluses on it, Ca++. So it binds calcium when present. So that implies calcium's not always there, but when it is there, it will bind it and pulls tropomyosin off or away from the actin, the binding site on actin. Off of the actin binding sites. So on actin, you have a spot that myosin can attach to. That's a binding site. And if calcium is available, calcium will attach to troponin. Troponin will pull tropomyosin out of the way and expose that binding site so that myosin can attach to it. You're going to notice that I'm repeating myself 5 million times today. You guys need to do the same, okay? Because it'll make more sense the more you'll repeat it the first time and you'll get a couple little details. And you repeat it again, you start to get it, and you're like, oh, that's what that meant. And then finally by the 5 millionth time, you'll be like, aha, I got it. And you're going to ace that exam, right? Yes. So when calcium is present, troponin changes its shape and it pulls tropomyosin out of the way, because you guys remember proteins can have different shapes, right? And when something binds to it, sometimes it changes its shape. That's what's happening here. So pulls tropomyosin out of the way to expose the actin binding site so that the myosin head can bind to actin. So if we look at the pictures down here, okay, so I think myosin kind of looks like golf clubs. So it kind of has like the funky golf club head here. Okay, with the stick part of it that you hang on to if you're going to play golf. And actin is the double beaded strand here. So we have actin here, and this one is myosin. Now, myosin isn't just one head, but usually when we're talking about the physiology, we're talking about what one thing is doing at one binding site. But if you look at this filament, can you guys see how there's many myosin heads sticking off of it? Okay, and if you look at the actin, There's actually, every time you see one of these troponin molecules, to me they look like little blue snowmen. Okay, so on the troponin, calcium is going to bind to that. The little green strand is the tropomyosin. If we pull tropomyosin out of the way, myosin can now bind to actin. Okay, so also, myosin is the thick filament, actin is the thin filament. Okay, so you might have to come up with your own way of trying to memorize how how you want to remember one versus the other. Okay, and let's see what else. If we talk about a sarcomere, so we're going to come back and talk about these guys some more, but that's essentially what the two protein strands look like. And we're going to learn what's called the sliding filament theory, because these are myofilaments, okay, and they're going to slide across each other, which will allow us to contract or shorten our muscle. Okay, so we look at the term sarcomeres. These are our little train cars lined up against each other. These are discrete segments of the myofibril. So discrete meaning small segments. And when we look at the sarcomere, we can see how myosin and actin actually interact with each other. It has Z discs. So the Z disc is the ends of the sarcomere unit. So a sarcomere is from one Z disc to the next Z disc. That would be one sarcomere. And then that Z disc to the next one would be another sarcomere. Okay, and there will be tons of sarcomeres end to end like a train car along the whole length of the muscle organ. So Z discs are specialized proteins that serve as an attachment site for thin filaments. Who's the thin filament? No. Thin is actin. Okay. So you could think actin, thin. Actin is a shorter word than myosin. Myosin's a big word, so it's thicker. I don't know. If that works for you, use it. So specialized proteins that serve as attachment sites for the thin filament, meaning actin. And the Z discs are perpendicular to the myofilaments. So they are perpendicular to the myofilaments. So what I mean by that is, if this is one Z disc and this is another Z disc, and I'll put another one here. So I'm actually showing you two sarcomeres because it's Z disc to Z disc. Okay, and if I'm saying that my myofilaments, actin and myosin, they go in this direction, can you see how I mean that it's perpendicular? It's going like this, it's not parallel. That's all it means. Z discs are perpendicular to the actin and myosin. So this will be actin, this will be myosin. This is an actin, this is a myosin. Actin, actin. Actin, myosin. So I think that'll help you guys kind of visualize this a little better. So actin attaches to the Z disc at both ends. Between the Z discs, myosin is suspended. So myosin's not attached to the Z disc. Okay. Now we're going to talk about the banding pattern. And there's a picture down here that helps you guys kind of see. So this is one sarcomere from one Z disc to the next Z disc. I'm going to scroll up just a little bit. And can you see this is saying one sarcomere to the next sarcomere. So this is one Z to the next Z. So this is one. So that line where they said sarcomere, they're actually showing you 1, 2, 3 sarcomeres lined up next to each other. Okay. So let's talk about what this banding pattern is. It says it's mostly due to myofilament overlap. So we use this to describe our picture, to describe the banding pattern. And what do I want to tell you? I don't know. I was all over the place with my notes. So the A band is a dark band. And you can remember A and dark because dark has the letter A in it. This will help. All right, so what it's talking about is if you were looking under the microscope or you were to shine light through this, if it's darker, it means more things are lined up against each other or on top of each other, right? So light won't shine through it as easily. So the A band is a combination of an overlapping of actin and myosin. So overlapping, so you have two protein filaments over each other. That's why it's darker, less light passing through. Overlapping actin and myosin. Okay. The I band is the light band, so more light can pass through it. And so look, I and light, I for I band. Nice, right? So this is the light band, and this would be actin only. So no myosin is in the I band region, okay? And so this is where the Z disc would actually be in the middle. So you're actually looking between two different sarcomeres if the Z disc is in the middle, because the Z disc is the end of one sarcomere, so you're looking on either side of where one came to an end and one is starting. Okay, and the actin is tethered to the Z disc. Actin is attached here, but it can still move around. So it's not like the, if you use the term like somebody had like a bunch of fence posts and say the Z disc is the big post that's stuck into the ground, this isn't stuck into the ground. Otherwise, your whole muscle couldn't move if it's stationary. Does that make sense? So the actin's attached to the Z disc, but it's not like the Z disc is stuck in the ground where it doesn't move with it, okay? Otherwise, you couldn't shorten in between each sarcomere, right? Okay. The H zone is where light passes through myosin only. So it's a lighter area in the middle of the A band. Because remember, A band had myosin and actin, but the H zone is just where there's only myosin. There's no overlap of the actin in that part. And then we have the M line, M for middle. This is kind of like a metaphase, right? M in the middle. All right. So this is a meshwork designed to keep the myosin in place during contraction and relaxation. So this is in the middle of the H zone. So meshwork designed to keep myosin in place whether it's relaxed or contracted. All right, so that striping, like you guys said earlier, is the striations. This is what gives you that striping pattern, okay? So the darker striping or area is due to less light being able to pass through it, or the myofilament, and the lighter stripe is the area that is due to more light being able to pass through it. And if you look at the A and the I band of that sarcomere down here, so let's see, we have, here's the A band, okay, which is not to Z disc, okay, because the A band was just where actin and myosin were overlapping, okay, and then this area here, so we have the Z disc is in the middle, we said, right, and the I band is actin only, okay, so can you see that some of these might change depending on if a muscle contracts. If we say these filaments are allowed to move across each other, okay, so which one is darker, do you think? The I band or the A band? If you just look at what filaments are overlapping each other. This one's I, this one's A. The A band would be darker, right? Okay, and then the lighter band would be the I band, which is what we set up higher where we set our banding pattern and what it looks like. And let's see, what else do I want to tell you? This area here would be an example of a relaxed sarcomere or a relaxed myofiber. So relaxed myofiber, one muscle cell, and also relaxed. If that's relaxed, then it means the sarcomere. That's an R, let's fix that. Relaxed sarcomere. Okay. And let's see. They also were showing you, if you go back and look at the difference where H zone is where myosin is only, so now you guys can see it, myosin was only from here to here, correct? So this whole area would be the H zone because there's no actin overlapping it. The M line was what went through the middle and kind of held your myosin kind of in place, right? Whether it was relaxed or contracted. And what I would say is draw a picture like this and see if you can recreate where the A band is, where the H zone is, where the M line is, where are the Z discs. So just do a stick drawing like that, especially when you get on the exam, but draw it a couple different times so you get comfortable about who is in what region. And that will help you guys kind of solidify the info in your head. So another thing I was going to point out is we're about to talk about something called sarcoplasmic reticulum. What other things did we learn about a cell that had something that sounded like something reticulum? We had endoplasmic reticulum. You had smooth or rough. Rough had the ribosomes. It made protein. Smooth had no ribosomes on it. It was more for lipid and detoxification. We didn't talk a whole lot about that one. But that's what sarcoplasmic reticulum. Sarco in the muscle. So it's in our muscle cells. Okay. And it's the little blue veiny looking thing right there. So for short, sarcoplasmic reticulum will be SR. Okay, if we look at the picture a little higher up, on this one, they're showing the sarcoplasmic reticulum. It's like the green, blue, and purple lines that are here like this. And it has like a meshwork that's going around all the different myofibrils. I'm going to erase that, but I just wanted to point out that that's the sarcoplasmic reticulum there too. And you're going to learn about what that guy does in a second. And as you guys are reviewing your notes, when you read about it later on in the notes, go back and look and see if you can pick it out somewhere else earlier in the notes. All right, so sarcoplasmic reticulum is, and here's another, I should have just scrolled down. There's a better picture of your sarcoplasmic reticulum. And again, they're using kind of a purpley blue color and the green striping there. So it's a sac-like membrane, which is just like what endoplasmic reticulum was, right? Sac-like membrane. Complex that wraps around the myofibrils. Okay, so this is where we start to learn about myophysiology, which is how a muscle contracts or shortens. All right, and what else do I want to tell you? So this is similar to endoplasmic reticulum. So I'm just going to do ER so you guys know endoplasmic reticulum. This is the important job of sarcoplasmic reticulum. It stores calcium. This is huge. So it stores calcium. So endoplasmic reticulum did proteins. Sarcoplasmic reticulum is storing calcium. And this controls the level of calcium inside the muscle, the muscle cell. Okay. So when the muscle is relaxed, there is no calcium interacting with troponin. Okay. And it interacts when if calcium is available, it has been released by sarcoplasmic reticulum. So it was stored in there, but let's say we go, hey, I want calcium, I want to contract a muscle. Sarcoplasmic reticulum will release the calcium. Calcium can bind to troponin. Troponin will then change its shape and pull tropomyosin out of the way. That exposes the binding site on actin, and then the myosin heads from the myosin filaments can attach to that binding site on actin. And then it'll pull on the actin fibers, and they slide across each other, and that's what shortens the muscle. So we will talk about all these details some more. I'm just peppering them at you over and over again, so it just will become like a fairy tale. You just know all the details of it, and you'll never forget it, right? So let's see. All right, and then if we're done contracting our muscle, what do you think we need to do with the calcium? We need to put it somewhere else. We need to get it out of that area on the muscle cell. We want to store it back in the sarcoplasmic reticulum till we want to contract again, right? So and we'll talk about how we do that. So let's do a little picture or diagram here. So I'm going to do a very simplistic drawing of what happens with the sarcoplasmic reticulum. So if I have here a one muscle cell, okay, so it'll have its nucleus and their nuclei are, off to the side, and technically it's skeletal muscle, so we could have like multi-nucleate, right? Don't forget. So this would be my if it's one cell, what's surrounding my cell? The plasma membrane, so it's a sarcolemma, okay? Inside of my muscle cell, I'm going to have sarcoplasm, right? The cytoplasm of a muscle cell, sarcoplasm, okay? And what I'm going to do is I'm not going to draw the mesh-like scenario that you see in that picture. I'm just going to draw a circle, and this is going to be my sarcoplasmic reticulum. So I'm going to put sarcoplasm, oh, I did the wrong one. That's plasm. Sarcolemma is what I meant to say, correct? Boop. Sarcolemma. Okay, inside is the sarcoplasm. And then I've drawn a sarcoplasmic reticulum, which remember is this mesh-like, veiny-looking thing, but it's not. In here, I'm just going to draw it. And what's inside? What do I store inside of my sarcoplasmic reticulum? Lots of calcium, right? So I'm going to put calcium inside. Hello, calcium. All right. So when we're going to learn these details in a second, but I'm going to have a motor neuron, because remember, I want to tell my muscle to contract. I want to pick something up. I'm voluntarily deciding to do this, okay? So I'm going to have a nerve that's going to end up stimulating it, and it ends up causing, we'll learn these details in a second, a voltage change on my plasma membrane. Okay, so we will end up getting a voltage change, triangle is change. Okay, so a motor neuron will stimulate my muscle cell, causing this voltage change. In my sarcoplasmic reticulum, I'm storing my calcium in it, okay? In order for the calcium to get out, we need something called a calcium gate. So I'm going to put calcium gates around it, and I'm going to make that another color just so it's easier to see. So I'll do red. So I'll do circles for calcium gates. Okay, so circle equals calcium gate. And then I'll do another color in a second. Let's do green. All right, so in order to open these calcium gates, to let the calcium out, we have to have the motor neuron stimulate the muscle cell. When that voltage change happens, it opens the gate. Now calcium can go where? Why does it want to go out of the sarcoplasmic reticulum? Think about regular simple diffusion. Area of high concentration to an area of low concentration. If you open that gate, calcium, you have high concentration inside of the sarcoplasmic reticulum, low concentration in the muscle cell. So it's going to flow through the gate from the sarcoplasmic reticulum to the cell. So calcium is going to go this way. If calcium is now available in my muscle cell, it can bind to troponin, pull tropomyosin out of the way, expose my actin, my myosin binding site on the actin filament. Okay, do you see where we're going with this? Keep repeating the whole story. I know it seems kind of dumb, but it will help lock it in, and you guys would do so much better on the exam if you do that over and over and over again, okay? So calcium gates are voltage dependent then. They don't open unless you get that voltage change. So the nerve cell, and we'll learn about that in a second, is causing the voltage change. This opens the calcium gate. Calcium now goes from high to low concentration into the muscle cell. Okay, once the calcium gate opens due to the voltage change, oh, I told you that. See, I'm getting ahead of myself. So when the contraction is done, we just said a second ago, what do we got to do with calcium? If we're done with our contraction, we don't want calcium in the muscle cell anymore. We need to get it where? Back into the sarcoplasmic reticulum so that we can cover up those binding sites and our muscle will not be hanging on anymore, and it will let the filaments go and stretch out, relax those sarcomeres, thus relaxing the muscle. Okay, so what we need is a pump to put calcium back in because it's still a higher concentration in the sarcoplasmic reticulum than it was in the muscle cell. And so if we have to pump it, it means we need what to do that? We need ATP or energy because a pump means we're going against its concentration gradient. So I'm going to draw in blue calcium pumps as triangles. So a triangle equals calcium pump, which then you guys know means you need ATP in order to do it. So now I'm done with my contraction. I'm going to put calcium back into my sarcoplasmic reticulum. Okay, so but before we can pump calcium back in, we need to close the calcium gate first, right? Because if we leave the gate open and we're trying to shove calcium back in, it's just going to flow right back out. So how do we close the gate? We need that voltage change again to go back to what we would call a resting membrane potential. So we changed, it was a voltage change that allowed us to open the gate. So in order to close it, we need to shut off that voltage change and put it back to normal. Okay? So in order to do that, what do I need to do? So in order to close the gate, we have to pump calcium back in. This is a cascade of events that occurs in a specific order. You have to memorize this. This is the beginning of myophysiology. Get it under your belt. I've told you all of that. Neurophysiology, of course, will be easier for you guys to learn once you get this. So we're going to finish how we make that voltage change occur again in a second. We have to learn a couple more terms, and then we come back to this. So we have to look at what is the nerve doing talking to this cell. Whether we get that voltage change or not, whether we then get it. So if we don't get a voltage change, will calcium leave the sarcoplasmic reticulum? No. If we don't get a voltage change back to normal, so stop that voltage change that had occurred, then we can't get calcium back in. Okay? So this picture here, what you guys should do after we learn about motor unit and the other information, this is showing you the nerve, the neuron cell, okay? It's showing how it interacts with the one muscle cell, and then it's talking to you about things that you haven't learned about, we're going to see in a second, and then you guys can see the striping on here of your sarcomeres, right? Here's a Z disc, and the other Z disc is hiding over here, not that one line that I just drew. There we go, okay? So, do I have info on that one? I do, motor unit. All right, so a motor unit is a single motor neuron or nerve cell. So don't just call it a nerve because you're not talking about the same thing then. So a motor unit is a single motor neuron or neuron cell or nerve cell and all the muscle fibers that it controls. So one nerve cell can control many muscle fibers. So one nerve do you guys remember looking at that neuromuscular junction for histology? This is what we're talking about now, okay? So, the neuromuscular junction is the specific site of innervation. Innervation means nerve going to it and telling it what to do of a single muscle cell by the neuron synaptic knob. So you guys saw before, this whole area here is a neuromuscular junction, okay? The end here is the synaptic knob, and it looks like it's directly touching your muscle. But there's actually a little bit of space between the synaptic knob and the muscle cell, and we call that the synaptic cleft. And a lot of stuff happens between the synaptic knob, cleft, and the muscle cell that we have to learn about. So the synaptic knob is that swollen end of the axon. So you have the nerve, the neuron cell coming down. It'll have multiple axons coming off of it. And then this is a synaptic knob. This is a synaptic knob. Here's a synaptic knob, bless you. The synaptic knobs have synaptic vesicles at their ends. And what did you guys learn about vesicles with other cells when we talked about cells? It's a membrane-bound thing that carries stuff, right? So we could do endo or exocytosis. In this case, we're going to do exocytosis of neurotransmitter. So NT for neurotransmitter. And with muscle cells, skeletal muscle cells, I should say, there is a specific neurotransmitter, and it's called acetylcholine, ACH. So ACH is the specific neurotransmitter for skeletal muscle cells. Okay. Let's see, what else do I need to tell you? So the synaptic knob is the swollen end of the axon. It doesn't touch, but it's very close. Inside the synaptic knob are the synaptic vesicles, the membrane-bound sacs that contain the neurotransmitter. And for skeletal muscle, it's what? ACH, or acetylcholine, okay? The synaptic cleft, this is that space between the synaptic knob of the end of the neuron and the muscle cell, that space in between. This is the narrow area between the synaptic knob and the motor end of the plate, or motor end plate, I should say. That's the motor end plate is the specialized area on the sarcolemma. Who has the sarcolemma? The muscle cell. So the motor end plate is on what structure? Muscle cell. Okay? So the motor end plate, specialized area of the sarcolemma containing the neurotransmitter receptor. So on the muscle cell is where you have the receptor that will recognize acetylcholine. Acetylcholine is released from the synaptic knob because of exocytosis of that vesicle that contains it. So let's see, do we have another picture coming up? Yeah, we have a better picture there. In the synaptic cleft, which says it's the narrow area between the synaptic knob and motor end plate, you have something called acetylcholinesterase. ACH, ACE. So what does ACE tell you again? It's an enzyme, and I told you usually the enzymes we're talking about are breaking down other things. So in the synaptic cleft, I just told you we need acetylcholine to stimulate our muscle cell, but we have a whole bunch of acetylcholinesterase breaking it down in that cleft area. The thing is while we want a contraction, we'll produce tons of this neurotransmitter and release it, and we'll have more of it stimulating the muscle cell than what gets broken down. But at some point we say, okay, stop contracting, I want to relax. And so then you stop producing neurotransmitter, and acetylcholinesterase will have time to catch up and break down the leftover ACH or acetylcholine. And then eventually there's no more acetylcholine in there, and you're no longer stimulating your muscle cell, so then you can relax. Does that make sense? Okay. I know all of you are like, yeah, that makes sense because you're saying it right now, but later when I think about this, I'm going to be totally confused. That's okay. That's why you're going to repeat it over and over again like a mantra. All right. So if I drew a cell, for example, just a generic cell, remember when we talked about sodium and potassium? Where do we have high concentration of sodium typically? Always on the outside of the cell. So you always have tons of sodium out here. Okay. And where do you have tons of potassium usually? Inside of your cells. And we learned before that you had, sodium potassium pump. You have leak channels that allow things to leak in, and we got to pump it back out against its concentration gradient, and so on and so forth. Okay. So here what we have is we have this nerve axon. So I'll put neuron or nerve cell axon, right? And it has these vesicles that it's exocytosing. And what did we say were inside of the neurotransmitter that we're releasing? ACH or acetylcholine, okay? And what we're going to do is there is what's called a motor end plate on the muscle cell, a receptor site that ACH will attach to, okay? If it attaches there, this will then open sodium gate channels. And I don't remember if you guys remember when I drew a cell before, we have these negative charged proteins. So overall, our muscle cells or any cell has an overall kind of negative charge on it. So if all of a sudden, if ACH attaches here and it opens sodium gate channels and lets sodium rush in because it's going from high concentration to low, correct? Okay, so if that rushes in, if we have a whole bunch of positive ions flooding our cell, what do you think happens to that negative charge that was on the cell? We've changed it. This is our voltage change that I just talked about a little bit ago. So when sodium rushes in, it changes the voltage on the cell. And all of a sudden, now you guys know more of the rest of the story. If sodium rushes in, we get a voltage change. What is sarcoplasmic reticulum now going to do? Release calcium. So if you had your SR right here with a bunch of calcium in it, we got our voltage change because sodium rushed in, then calcium will now go out. And now we can contract. Okay? So let's look at the details of that. I keep trying to feed you like little bits of the story, so keep adding to your story. Later, section off these little bits of the stories. Write them out. Write them in the sequence that it happens on. Then put them all together. And then take them apart again and look at the individual sequence and then stick them back together. Okay? So let's see. Motor neuron picture. Okay, so physiology of skeletal muscle contraction. This is when it's saying excitation, contraction, coupling. Excitation is talking about the neuron or the axon, the neuro part of it, because this is what's stimulating it, right? Telling us that I want to move my muscle. My brain said, I want you to move. This was voluntary, right? Contraction is on the muscle side of it, and it's a coupling. That's why we're starting to talk about the two things together here. So the nerve stimulus, at the neuromuscular junction, right? Neuromuscle junction, that makes sense. Okay, so we have what's called a somatic motor neuron. Soma, the root word of soma means body, and so it's just talking about somatic motor neuron means it's a voluntary thing that I'm telling it to do something, because we said skeletal muscle is voluntary. So these are action potential reaches the synaptic knob. And action potential is what is stimulating and passing the signal all the way down. It's almost like an electrical shock zipping down the cell, and it affects the whole cell, not just one little zip on one area. It's affecting the whole thing. Okay? This is part of what ends up allowing us to have that voltage change and send the signal down to the rest of the way. So somatic motor neurons, action potential, you'll see abbreviated as AP for action potential. Okay? That's where the voltage change occurs, is due to that. And now what do I want to tell you? So we said we have calcium enters the synaptic knob. So what it's talking about there is, if you go back to our picture here, calcium enters the synaptic knob. This is not the same calcium that's stored in your sarcoplasmic reticulum, because this is not inside of that cell at all. This is other calcium that is stimulating this coming in, okay? Exocytosis of neurotransmitter into synaptic cleft. That's this guy right here. We produce acetylcholine. We exocytose it from the synaptic knob. It goes into the synaptic cleft. It needs to go to the motor end plate, which is on the sarcolemma, right? Okay, when it attaches there, it then does what? Neurotransmitter diffuses across synaptic cleft. Neurotransmitter binds to the receptors on the motor end plate. That causes then sodium gates to open. Sodium floods the cell, causing the voltage change along the sarcolemma. Okay, so it's nice to see these pictures tell you the story, but sometimes you redrawing a simple picture on the side means more to you, and you can recreate that picture on the exam. Don't read the questions, just do your diagram really fast, and then that way you don't get confused and go, oh yeah, that answer sounds right. I must have had that wrong. Don't do that. Don't second guess yourself. Draw your little diagram, then answer the questions. Then you don't get sidetracked, right? By an answer that might sound good. Let's see, what else do I want to tell you? So acetylcholine is the key that opens the sodium gate. So if acetylcholine didn't attach to that receptor, you wouldn't get sodium rushing in because you wouldn't open the gate. Okay, so acetylcholine is the key for that. And let's see, skeletal muscle cell requirements. So I'm going to scroll down. If you guys need to, you just pause this, go back to this section, pause it so you can redraw some of those pictures for yourself. You don't need to draw it the way I drew it. Draw it in a way that makes sense to you, okay? So the motor neuron stimulates the muscle cell at the motor end plate, right? At motor end plate. After you guys like reread these notes 5 million times, you'll be like, she repeated herself like 5 times there. Yes, I did. Because you have to get this. All right, the muscle cell propagates an action potential, an AP, right? So this is a voltage change. Triangle means change. Due to what? What caused that voltage change? What allowed it? Due to sodium ions rushing into the cell. Okay? That spreads along the whole sarcolemma. Then the voltage change in the sarcolemma opens the calcium gates in the sarcoplasmic reticulum because they're really close to each other. Because if you guys went back and looked at this picture, can you see, here's your sarcolemma, the cellophane plasma membrane that wrapped around it, and see how close it is to the sarcoplasmic reticulum, the little veiny structures, right? And can you see how it wraps around the myofibrils all over the place? This is what allows it to stimulate everything like all at once. It's not like a delayed reaction where it takes a little while for that end of the cell to get the message. It's done, okay? Everybody gets the message quick because of that. And let's see. So calcium floods the sarcoplasm. It binds to the troponin complex. Troponin complex pulls tropomyosin out of the way, causing the cross-bridge cycling. And the sequence of events of cross-bridge cycling are dun, dun, dun. All right, let's learn this puppy. You guys already know most of the story. Now you just have to see it a little bit, okay? So, okay, let's see. There is no change in length of myosin and actin. Those fibers don't change length, but they slide across each other. That's the sliding across each other is what shortens the sarcomere, thus the contraction or shortening of the muscle. Okay, so actin and myosin don't actually shorten. That protein stays the same length. It just slides over each other. That's what shortens things. So, all right. It says calcium is binding. So we're looking at the sarcomere cross-bridge cycling. We're looking at 3A here. It says calcium binding. That means the sarcomere motor neuron had an action potential. It released acetylcholine, which attached to the receptors on the sarcolemma. This allows sodium gates to open and flood into the cell. The cell has a voltage change, which is felt along the whole muscle cell. And this causes calcium gates to open on the sarcoplasmic reticulum. Calcium is then released to the muscle cell and attaches to troponin, the little snowman guy. Troponin is on the thin filament actin. This causes a shape change that pulls tropomyosin out of the way, which was your thread that was covering the myosin receptor site, right? So it exposes the myosin binding site on the actin. Once this is exposed, the myosin heads, those little golf club guys, attached to actin. It's kind of like a magnet or like a tractor beam. It's like, bloop, and he sucks onto it and he's attached. Okay? He's not letting go yet. Let's see what else I'm going to tell you. On the picture here, just so that you guys know, these little white snowballs are the calcium ions. You have your little snowmen, which were the blue ones. That's troponin. And then you had the green thread, and that was tropomyosin. Tropomyosin will get pulled out of the way, and actin are the little red beads there. So you could think of it, Sherry likes to call them olives. I kind of think they look like cranberries. In the previous book, they were blue, and I called them blueberries. So if you want to think like the flower end of the cranberry, the little hole, that's where you're exposing. So tropomyosin was covering that up. When this gets moved out of the way because calcium was available there, you got that little flower spot open so that the myosin head can attach there, okay? So myosin, here is your myosin guy. He has the little golf club head on it. Okay, so let's see. When myosin has attached, when the myosin head attaches, ADP and inorganic phosphate get knocked off. So what do you guys know about ADP plus a phosphate gives you ATP, right? So ADP plus inorganic phosphate gives you ATP. Or you could break ATP down into ADP and a phosphate, correct? Okay, so when myosin head attaches, so now we're looking at this picture here, 3B, cross-bridge formation or attachment. It's showing you that the myosin head is now attaching because that myosin site got exposed on actin, and it's showing you that the ADP and phosphate is going to get kicked off when it attaches. So that's what this picture is showing you here. ADP and phosphate go by. When ADP and phosphate pop off, the myosin head has a shape change, and all of a sudden it bends or flexes, or you could think of it like a ratchet. It's like, okay? So as soon as ADP and phosphate pop off, it ratchets it in. Which direction do you think it's going to ratchet actin? Towards the center. Remember, actin is attached at the Z discs, and if we have myosin in the middle, so these are this picture here, so you have your Z disc here, you have actin, and myosin's in the middle. And I'll draw myosin a little thicker because remember, he's the thick filament, right? Myosin has the little head there attaching, okay? And if he ratchets in, we're sliding the filaments over each other, he's pulling actin towards the center. M for middle, remember our M line? He's pulling him into the center. This is a Z disc, Z to Z, this is one sarcomere. Okay? So, when it does that ratchet or bend or flex, however you want to think about it, I like ratchet because it makes you think like zip, right? It sucks it in. This pulls the actin toward the middle of the sarcomere, and this is called the power stroke. So power stroke. That's the ratchet part. Sucking it into the center, pulls it in. Like tug of war if you want, right? So now ATP comes in. So this is this next picture. Okay? When ATP attaches to the myosin head, it makes myosin release actin. So ATP attaches to myosin, it lets go of actin. Okay, and then it resets the myosin head. So it not only lets go of actin, but it also recocks the head back. So this was that flexed position. Now it's going to go back into that position where it's stuck back again. So it could re-ratchet, right? If it stays stuck in the ratchet position, it won't be able to grab onto actin and pull it in anymore. So it has to reset. So ATP attaches, myosin lets go of actin, and it resets the myosin head back. Okay, so ATP is needed for the detachment and resetting of the myosin head. This will happen over and over again, all of those stages in that order, and thus overall, so this just happened in one little spot. Do you think it moved your sarcomere very much? No, but remember, on the myosin filament, you actually had tons of little myosin heads sticking off, so it's like, but more smooth, right? And you're sucked in and you're contracted. And then everybody says, oh, we're done with calcium and we have ATP present, we're going to let go. And the cell relaxes and lengthens back out again, right? Okay. So you guys remember that actin is attached to the Z disc. So if actin is pulled to the center, so are the Z discs. This is why I said Z discs aren't planted into the ground like a true fence post, because the Z discs are approximating or coming together because of the filaments sliding over each other. So the whole Z disc is going to come in closer, right? Because actin and myosin are overlapping more. Okay? So let's see. Actin didn't shorten, right? Myosin didn't shorten. They just overlap, so they slide across each other. That's why we call this the sliding filament theory. This happens to all of the sarcomeres of the myofibril or muscle cell. So the muscle cell or muscle organ overall shortens and contracts. And let's see, what happened to the I band? So you know the A band shouldn't have shortened, right? Right, because that's the, actin doesn't shorten, A band doesn't shorten, right? That stayed the same. But what about the I band where actin and myosin overlap? Did that change? Yes. Right? So I bands are pure actin. As the sarcomeres shorten, myosin and actin are overlapping. The I band shortens. So it's where you don't have actin and myosin. It's just where the actin is. I lied to you a second ago, didn't I? And you guys all fell for it. I said it was where it was overlapping. It's not. It's just where actin is only, right? A-band is overlapping. All right. Mull it, think about it, review it. When we come back, I will, the H-band, let's see, where's our H-band? So what are we talking about on H-band? H-band passes through myosin only, so H-band would change, right? H-zone? Because if just this area is H, right? Just where myosin only is? So that would change if they're overlapping each other, correct? So play with this idea before we come back on Thursday about what changes on, are there changes with A, I, H, or M on the banding pattern when it contracts and relaxes? And I'll redraw the picture. I'm going to fill in verbally the notes for the power stroke, resetting the myosin head, removing calcium. But I want you guys to think about that part because we've already said it verbally a couple times. But I will fill in these notes, so when we come back, we'll briefly review everything that happened there, and then we can talk about the rest of that story and rigor mortis. Who doesn't like talking about rigor mortis, right? All right, awesome.